And her presentation is entitled Development of a Taqman Assay for the Detection of Groundnut Bud Necrosis, Orthotospovirus for Quarantine Screening. Anushi, thank you very much for being here today.
Yeah, thank you, Dr. Kamrat. I'm Anushi Deraniyagala from University of Georgia. Today I'm going to present a part of my master's research, development of a Taqman assay to detect quarantine pathogen GBNV. This is the overview of my presentation. First, I will talk briefly on plant quarantine and different diagnostic techniques. Then I will talk in detail about the development of Taqman multiplex real-time qRT-PCR assay and end my presentation with some conclusions. During the recent years, exchange of plant materials has been increasing. The two main reasons for that are the exchange of plant materials allows accessing the best plant trait, and also it helps in exchanging germplasm worldwide. Exchanging plant materials is really important, but at the same time we have to make sure that there are no exotic pathogens are introduced with these plant materials. Here comes the importance of the plant quarantine. Plant quarantine measures allows the exchange of plant materials while safeguarding agriculture and natural resources in a country. In a quarantine procedure, detection is really important, which is also the very first step. For the detection of pathogens, there are several diagnostic assays that we can use. Among these diagnostic assays, we decided to focus on developing PCR-based techniques to detect GBNV because PCR techniques are widely used and they are relatively specific and sensitive. When it comes to real-time RT-PCR assays, there are different types. We also decided to go for developing a Taqman assay. In the SYBR Green assay, the dye SYBR Green is involved. In the Taqman assay, the probes are involved. The Taqman assay is highly specific and sensitive due to the involvement of probes. We decided to go for the Taqman assay. I would also like to tell some information on GBNV, which is also known as peanut bud necrosis orthotospovirus. This is an orthotospovirus and currently restricted to the regions of Southeast Asia. Despite the implementation of different management strategies, still sometimes this pathogen can cause a significant problems. Why this is important as a quarantine pathogen? This can infect several leguminous and solanaceous hosts such as peanut, potato, bean, and tomato, and also this is transmitted by thrips. This is a map showing the distribution of the hosts within the United States. The vectors are also present within the US. Because of the presence of hosts and vectors, if GBNV is introduced, it could get easily established. With that, the objective of my research was to develop a nuclear-based diagnostic technique to detect quarantine pathogen GBNV. Proceeding to methodology and results, the first step is the sample collection. Since this is a quarantine pathogen, we received peanut leaf infected with GBNV from India. We received from 2 locations, which are from 2 isolates. After the sample collection, we had to confirm the presence of GBNV. First, I have designed the primers targeting GBNV movement protein and GBNV nucleocapsid protein. We observed successful amplification, which resulted amplicons. Then these amplicons were cloned and sequenced. With the help of these sequences and the nucleotide blast tool in the GenBank, the presence of GBNV was confirmed. After the confirmation, here comes the important part, development of Taqman-based real-time qRT-PCR assay. This starts with designing primers. The primers were designed targeting regions around 100 to 200 base pair in size, because this size is size is preferred in Taqman assay, and then the protocols were optimized. The protocol optimization involves the optimization of reagent concentration and also the cycling conditions. Final step was the standardization. For standardization, several controls were included. One of the controls were the internal controls. Here we included primers targeting host plant genes. The other important control is the negative controls, which involves healthy peanut RNA and peanut leaf RNA, which were infected by the closely related other orthotospoviruses to GBNV. In addition to that, DNS-treated RNA versus non-treated RNA, and then reaction mix with non-reverse transcriptase and without reaction mix with non-reverse transcriptase and without reverse transcriptase were also used. After that, this table shows the genomic regions targeted for the assay. There were 3 different fluorescent dyes in each probe specific to each region. This shows annealing temperature and the amplicon size for each primer pair targeting each region. The regions targeted were movement protein, nucleic acid protein, and 1 plant gene as an internal control. After optimization of the cyclic conditions and region concentrations, next comes the standard curve construction. Standard curve construction is really important because it gives the values such as efficiency and R-square values, which are important to know about the reaction, and the efficiency and the efficiency of the reaction, which allows us to further optimize if the efficiency or else R-square do not meet the optimum values. The standard curve construction starts with the total RNA extractions. First, total RNA was extracted from peanut leaf samples infected, and then 2-fold serial dilutions were prepared, consisted of 15 different dilutions. Finally, the fluorescence data were analyzed to get the values such as efficiency, R-square, and standard deviation with the help of Bio-Rad CFX Manager software. These graphs shows the amplification curves that were obtained during standard curve construction. We found that our assay is sensitive enough to detect GBNV nucleic acid protein and movement protein in a total RNA amount as small as 0.098 nanogram. These are the standard curves resulted. The efficiency values should be within the range 90 to 110. The coefficient of determination should be within the range 1.0 to 0.98. The slope values must be close to each other. In our assay, we found that the efficiencies as well as the R-square values are within the expected range, and also the slope values. With knowing that the singleplex assays are optimized, we proceeded to multiplexing. It was also successful. We didn't observe any cross interactions. If I talk about the difference in between multiplexing and a singleplex reaction, in a singleplex reaction, we include 1 primers and probe set targeting 1 region in 1 vial. During the multiplexing, in 1 vial, we include all the primers and probes targeting different regions in 1 vial. So there's a higher chance to observe cross interactions since all the amplifications happens at one time, at once. So we didn't observe any cross interactions, but we still need to know about the efficiency of the multiplex reactions. Here what we did was we obtained CQ values for each region in both reactions, multiplex and singleplex reactions, and we compared the CQ values in between both reactions. When we compared these values using non-parametric Wilcoxon sign rank test, we found that there is no significant difference between CQ values when it comes to viral genomic regions, GBNV nucleic acid protein and movement protein, but there is a difference for the internal control. With that, I would like to discuss few facts. Primus probe sets designed are highly specific to GBNV, and we confirmed using healthy peanut RNA and closely related orthotospoviruses infected plant RNA. These orthotospoviruses were TSWV, GRSV, and TCSV, which are already present within the US. The singleplex assays have efficiencies over 90%, and multiplexing is possible at 53 Celsius. There is no significant difference in CQ values in singleplex and multiplex assays when it comes to viral specific genes. With that, I would like to state some future plans. We are trying to validate the assay using more field samples. We are also planning to do absolute quantification and calculation of efficiency in multiplex assays. And then finally, we hope to transfer the technology and to implement at the port of entry. With that, I would like to thank my committee members, Dr. Sudeep Bag, Major Advisor, Dr. Kulbert, and Dr. Dion. And also, I would like to thank lab members and other collaborators and funding agencies. And also, I would like to thank Dr. Kamret for organizing this. Thank you.
Thank you, Anushi. Very nice presentation. And we have one question already. And is the question from Shyam Tuluri? It says, is GBNV seed transmitted? If so, what's the vector for the virus transmission?
GBNV is an orthotospovirus, so it is not seed transmitted.
Okay.
The vector for the GBNV transmission is thrips species, and other than that, for greenhouse experiments, it is possible to transfer through mechanical inoculations.
Thank you for that.